Aperture Therapeutics is advancing APRTX-003, a candidate that abandons the wait-and-react protein model in favor of a precision RNA targeting strike. By acting directly on the genetic instructions that drive protein production, this strategy introduces a new level of precision that could meaningfully expand what is possible in neurodegenerative treatment. The core innovation of APRTX-003 lies in its identity as an antisense oligonucleotide, ASO for short. ASOs are short strands of synthetic genetic material engineered to bind to a gene's mRNA, the intermediate blueprint the cell uses to produce proteins. When APRTX-003 binds to the mRNA for the harmful enzyme MMP9, it marks that template for destruction, ensuring the toxic protein isn't produced, while traditional small molecule drugs attempt to inhibit proteins that are already circulating and causing damage. ASOs intervene one step earlier. By going upstream, this approach seeks to limit the creation of harmful proteins altogether, reducing the pressure on the nervous system to manage them once they accumulate. Given how vulnerable the brain and spinal cord are to cumulative injury, preventing damage at its source is widely considered more promising than attempting to reverse it once neurons are already compromised. The target of this therapy is matrix metalloproteinase 9, MMP9, an enzyme increasingly recognized as a significant contributor to the biological processes that accelerate ALS. Studies have shown that people living with ALS, and in some cases other neurodegenerative conditions, exhibit elevated MMP9 levels and heightened enzyme activity. Previous attempts to inhibit MMP9 using small molecules or antibodies failed because they lacked the necessary specificity, often interfering with other essential enzymes and causing dangerous side effects. Aperture's founder and CEO, Martin Jacko, has emphasized that although MMP9 has long been supported by strong biological evidence, earlier attempts to target it with small molecules or antibodies were hindered by poor specificity and unintended interactions with other essential enzymes. In ALS pathology, elevated MMP9 activity has been associated with mechanisms that intensify neuroinflammation and contribute to motor neuron loss. Preclinical studies targeting this enzyme have produced encouraging results. Research indicates that lowering MMP9 levels can slow motor neuron loss, delay the decline of motor function, and extend survival. Crucially, Aperture has validated these findings using a humanized mouse model, one engineered to produce the human form of MMP9. Aperture's model adds compelling support to the idea that MMP9 represents a biologically meaningful point at which ALS progression might be slowed.